Thank you. Okay, I'm Behruz. I want to talk about plants, unlike other people so far that have been talking about animal cells. I come from Lund University in Sweden. My supervisor is Henrik Jansson, who is a professor of computational morphodynamics. It's a new interdisciplinary field in Cambridge, in Sainsbury Lab, and this will be a summary of the work that we have done during last 3-4 years. And actually, it's all about mechanics and how much we can get out of that and to be used for other components of the system in a plant to produce these phylotactic patterns. And this work is based on one published paper and 2 other papers that are in the pipeline. The outline will be, I will talk about a few. Yes. Okay. This is not. Okay. I will talk about a few general concepts, and then I will define the main question. Then I will introduce you with our mechanical model, the implementation and the validation that we have done. Then we talk about the stress feedback model that we have applied to our, added to our mechanical model, and the motivations for this stress feedback model, and a kind of analytical analysis on this kind of a stress feedback model based on energy minimization. Then we have added strain-based growth to our model to achieve really large deformations, and recently we have added a full 3D investigation. Okay, the most interesting part of the plant is this tiny part, which is very small, around 100 micrometers, and all the patterning of the plant happens, initiates from here. And the rest of the lifetime of the organs are just growing. And all of these nice phylotactic patterns initiate from here, following a kind of auxin patterning that happens on top of this tiny pool of stem cells that is called meristem. And these organs, some of them become leaves and some of them flowers, but they all follow the same pattern. Yeah, actually, the cell walls are the main responsible component in the rigidity of the plants. And this rigidity comes from the fiber networks inside the plant cell wall. And another very important thing is this turgor pressure that, unlike the animal cells, is around 3 to 5 atmospheres, and you can imagine that is huge. And another key concept is that the epidermal cell layer, which is much thicker than the other cell walls in the plant, plays a very important role in patterning, because you can assume that this pressure can be cancelled out on the sides of the internal cell walls, whereas, which is not the case for the epidermal cell walls. And this, the importance of 3 first cell layers is not just the mechanics, but it's shown via many experiments that there are many biochemical reactions happening specifically in these layers. And so the cell walls are the only rigid part of the plant, and the rest is mainly liquid. Yeah, and if you look at the structure of the cell wall, actually the most rigid part of it, which is the fiber network, there are a lot of players, but the main components that are responsible for the rigidity are these fibers that are as stiff as steel. And they have, but the distribution of these fibers can be random or completely anisotropic. And it depends on the microtubular network, cortical microtubules that are tethered to the plasma membrane. And there is a very nice machinery here that a motor protein that moves along the cortical microtubules, and the other side of the plasma membrane builds up these deposits, these fibers. So if you can visualize these microtubules, you can predict that the fibers that are deposited are following this direction. And a very, very nice feature, actually it's not that easy to visualize these fibers, but this is not the case for microtubules. There are many nice GFP markers for visualizing this microtubule network.
Doesn't the microtubule network turn over in time?
Yeah.
Whereas once you have laid down.
Yeah, they are very, very dynamic, and I will talk about that. Actually, the directionality of microtubules is variable via space and time. So if you look at the meristem from top, you can see that they are randomly distributed on the central zone on the meristem, whereas they are very organized circumferentially around the meristem. Actually, as these new organs around the meristem form, the organization of microtubules around the new appearing organs change via time. So they are not constant. And actually, from them we can predict how the new layers of the fibers are getting deposited, but we cannot predict the past because other layers could be deposited earlier according to some other distribution of microtubules. But it has been shown that the latest layer of fibers are the most responsible part in this network of fibers, because during the growth they get diluted, and actually they get deposited from one side, but they will be stretched, and the thickness of the wall stays constant. Yeah, this is a very nice paper if you are interested in the relation between stress and microtubule directions. And another very nice feature of microtubules is that there is a very, very strong correlation between the stress directions that can be predicted from the calculations. I mean, analyzing the surfaces from the geometry and the shape of the organs in plants and microtubular directions. On top of the meristem, where the stresses are isotropic, you can see a very isotropic distribution of fibers, whereas around the wherever we have an anisotropic curvature, we have also anisotropic distribution of the microtubules. And this is the extreme case is in the boundary between the new organ which is formed and the central zone of the meristem, which has the negative curvature in one direction and positive curvature in the other. And it has been shown in many, many different experimental and analytical papers that there is a very, very strong correlation between the microtubules. This is an example of the a completely different domain in the plant. It's on the leaf, actually pavement cells that are very wavy shapes, and you can see the macrotubules, and this is the result of a simulation, finite element simulation that shows that actually, and here again, this is the simulation and the orientation of macrotubules. Now, not only the directions, but also the concentration of the macrotubules correlate well with the stress. So this having these in mind, we ask this question: that what's the proper candidate for regulating the macrotubules in a purely mechanical point of view? If you look at the structure of this, is the a very schematic shape, which is because many parts of the plants include the stems, and the rest are really kind of dome shapes that are located on top of the organs. If you look at a cylinder, a perfect cylinder, we can see that the stresses, we can show that analytically that the stresses are mainly circumferential. And of course, there are longitudinal stresses, but they are mainly circumferential. In the experiments have been shown that the growth happens axially, and the plant grows axially along the longest axis of the cylinder. Having this information, it has been suggested there has been 2 points of view. One suggesting that the stress regulates the direction of macrotubules, or the principal stress, and an older theory that relates macrotubules with the perpendicular direction to the strain. So if we take a strain as a cue for the growth, and the perpendicular direction of the maximal strain will be the regulator of macrotubules. We have compared these two. We wanted to compare these two, so we developed a kind of a very basic anisotropic model, which has been according to the, modifying the well-known Saint-Venant description for the strain energy, and we have penalized that according to the equipartitioning of the energy in different directions with this term, which includes the anisotropy vector and some difference between the material properties in the longitudinal and transverse directions. Yes?
Here. I'm confused. You're talking about strain in the cell wall now?
There are strains and stress.
Yeah, but isn't the cell wall intercalating new material? So it's not as though you have a static material that you're straining, but you're actually adding new material, aren't you?
Yeah, there is of course new material, but mechanics is very fast, and we assume that mechanics is in equilibrium. I mean, the material is always in the mechanical equilibrium. This growth is much slower than the mechanical interactions between components.
But how do I define strain if I don't have some reference configuration?
You have a resting shape, and you have a stretched shape or deformed shape, and you can always compare these two. Yep. Okay. So we have these we have these material constants and anisotropy direction that can, in addition to strain, can give us the value for the mechanical energy, and then we can take the derivative respect to a strain and calculate the stresses on different elements. But we can use either the different elements for describing the cell wall. We can use shell elements that are more advanced elements. They have some kind of thickness, or planar elements that lack the bending energy, but they are computationally faster. So we performed a comparison between these two because we assume that these elements are can include also the bending energy, and they can describe the material better. And we did a lot of comparisons in plane or out of plane. I mean, in three-dimensional. And in plane.
Sorry, are these only poles on the surface?
In plane is only in the surface.
Right. And
Sorry.
And the shell element there,
The planar elements, they don't have any thickness.
yes.
And when there is a junction between two elements, there is nothing to stop them from, I mean, at the junctions you have no bending energy. But shells have thickness, and when they meet each other, they can really, I mean, if you bend a junction, which is the intersection between two shell elements, you have to deform them. Otherwise, you cannot. Which is not the case for the planar elements. So we did in plane tests that they don't have any, there's no occasion in plane tests to have some kind of bending, so they were agreed ideally. I mean, there is no difference between shells and plates. And for the out-of-plane tests, we have pressurized different shapes, a square, which is pressurized from below, a dome shape, some kind of a part of parabolid that has been pressurized, and a kind of shape which represents a kind of a part of meristem with a new organ, or another part which can be of interest, which is the boundary between two organs, and has a negative curvature in one direction and a positive in another. And as long as we have positive curvature everywhere, we couldn't see a considerable difference. The only case is when we have a kind of saddle shape which has a negative curvature in one direction and positive curvature in another direction. And you can imagine that in those cases, the bending energy can play a very important role. But these are just a few parts of the plant structure. So, for the next test, we asked are stress and strain can be treated the same? I mean, are these signals equivalent when we have an anisotropic material? The answer is no. When you change the, I mean, here I have represented a kind of patch of elements that have been under some kind of load. This load can change from isotropic load to perfectly anisotropic load that can be represented by 0 and 1 for the value of force anisotropy, and the other axis is the stiffness ratio that can represent the degree of anisotropy of the material. And I have represented that with these lines. Here the material is isotropic and the force is also isotropic. Here the material is isotropic, whereas the force is uniaxial and perfectly anisotropic. Here, it's maximum anisotropy for both force and material, and here we have anisotropic material with isotropic force. And if we look at the directionality of the stress and strain, when we change these two, we come up with two regions, two distinct regions, in which in one of them these stress and strain are perfectly perpendicular, and in the rest of the regions they are aligned. And if you think about a geometry like this, a shape like this, you can imagine that according to the difference of the curvatures, you can end up with different anisotropy properties for the stress. And if the material is also in some places isotropic and anisotropic, you can have different scenarios. In the central zone, we can imagine that the material is isotropic because the distribution of microtubules is isotropic, and the forces are also isotropic because in the central zone the stresses will produce a kind of isotropic field. Here I have shown the peripheral zone a bit away from the central zone, where we have more stresses circumferentially, and we have almost anisotropic stress and also anisotropic material, because the alignment of microtubules are anisotropic. And also the same goes for the rest of the plant. So in different places we can have different scenarios. So we have applied our model to a kind of cylindrical geometry, and we have had a kind of anisotropy value for the material around 3. So the material has been 3 times stiffer in the circumferential direction than the axial, and we have pressurized it, and the direction of anisotropy of the material has been circumferentially. Yes.
Are you discretizing the material property?
Pardon?
Are you discretizing the material property?
Each cell has its own material property.
Okay.
You can have different material properties in different cells.
So each cell have its own.
Yeah. But it includes many elements. But all these elements that are in one cell, they have the same material property. And also, when I say material property, it includes both the direction and the degree of anisotropy. So we pressurized it, and we observed that according to having 0.5 stress anisotropy for a perfect cylinder, we end up somewhere around here, and the microtubules, I mean the direction of anisotropy and stresses are aligned, and they are also aligned with the perpendicular to strain. So it includes both theories, perpendicular to strain and stress. But then we compared if we apply feedback from perpendicular to a strain, I mean here, to this microtubular alignment, and also a stress, what will happen? Here is the result of applying the stress feedback to the, connecting these two distributions. I mean, we can always evaluate the stress anisotropy, and then push the material anisotropy vector to become aligned with this stress anisotropy. And we didn't see any change. Whereas for the other case, they switch and choose the axial direction. I mean, the anisotropy vectors, they become aligned. So although these two theories can describe this kind of static direction situation, but when we turn on the feedback, this assumption fails. And here I have shown that why this is so. When we show the principal stress direction and perpendicular to a strain versus anisotropy direction, one of them has a positive slope and the other one has a negative slope when they are aligned. They are all aligned here, and this can show a stable or non-stable fixed point of the system. We moved on and added adaptive anisotropy to the system so that the elasticity of two directions of the material can adapt to the stress anisotropy. Then the total amount of these elasticity is constant all the time. This is a very simplified assumption because we can assume that we have constant amount of fibers all the time, and if we distribute them uniformly or align them all in one direction, we should reduce the elasticity in one direction and add it to the other direction. So this very simplified assumption that we have made. And you can see that when we change the stress anisotropy in our model, we adapt the longitudinal and transverse anisotropy to the stress anisotropy via a kind of nonlinear fashion. This is a very ad hoc test that has been motivated by this kind of comparison between shapes. And we found a very interesting result. In this kind of geometry that has a anisotropic curvature, I mean it's a kind of parabolid shape, we can get a circumferential pattern of the stresses. We can have a kind of zonation that can be very interesting for other biological processes, in which in one region we have stresses and strains aligned, and in the peripheral zone the strains are radial, whereas the stresses are circumferential. And we get the highest growth rate in the peripheral zone, and we get the stiffest material, I mean, the most anisotropic material in the peripheral zone also. And we did this test, we repeated in a more complicated shapes, and we got more or less the same results. And this is very interesting because many models that don't include mechanics at all work with this kind of radial zonation. And but for these kind of radial zonation models to work, they have to anchor something, I mean, to the center in order to work. I mean, it's very difficult to produce this kind of radial zonation out of nothing. So it suggests that we can use this kind of shape information that is reflected to this kind of zonation properties of the stress and strain signals, and be used in these kind of models. And also I've shown a kind of experimental picture, which is a measurement on the shoot apical meristem, and you can see that the highest growth rate in the peripheral zone correlates very well with our simulation. Okay, inspired by these results, we wanted to go forward and ask two questions. What more do plants gain from the stress feedback? And can they use it for directing the plastic growth also? Because for elastic deformation is a tiny amount, and it's below maybe below 10%, maybe 6-7%. So this is not enough to create pattern formations. So we need the plastic growth model to add to our model. First, the result of a stress feedback. One very clear result of this model is that it amplifies any kind of anisotropy in the shape. So if you start from a kind of isotropic shape, it does nothing. The red shows the result of a stress feedback model, whereas the white is the result of perpendicular to a strain or isotropic material. They both show the same behavior. And but as soon as you have a kind of anisotropy in the shape, a stress feedback amplifies it all the time. And if the shape is more and more anisotropic, the amplification will be more and more. And this is very favorable for the plants because they like the anisotropic growth. Another aspect is that, okay, we normally, in all of the finite element models, we minimize a kind of energy respect to a strain. But we have two additional variables in our energy now, which are the anisotropy direction and the degree of material anisotropy. Can we use these two also? Starting from a linear energy, we can parameterize it in a constant stress field, assuming that the stress field is completely constant. And now we rotate the material, so and we measure the energy. And assuming that the stress can be isotropic or anisotropic, and the material can have different degrees of anisotropy. And by changing the direction of anisotropy, we ask if there is any, and changing also the degree of anisotropy of the material, we ask if there is any minimum for the energy or not. Because if you assume that one cell is located somewhere, actually the geometry defines the stress field that is applied on this cell. And if it decides to rotate the anisotropy vector or the degree of anisotropy, change the degree of anisotropy, by changing this, it cannot change the external stress field which is applied on this cell. So by this parameterization, we had this kind of, in a linear energy model, we had principal stress values and the direction of anisotropy, relative direction of anisotropy and maximal stress, and also two values for the elasticity, E and EM. And then by taking the simply derivative to theta, the direction of anisotropy, we can see that there is a clear minimum for the energy at 0 for the theta, and it has a maximum at 90 degrees. So if the material is anisotropic and the stress is also anisotropic, the minimum energy happens when these two are aligned. And now we can set this value of theta to 0 and ask if we can take another derivative respect to the ratio or the an anisotropy measure of the material, which is kappa here. And if we put this kappa and take again another derivative respect to kappa, we have another expression, which is not, if I put it equal to 0, I cannot find an explicit a simple equation between kappa and alpha, but it is clear that at least for 0 Poisson ratio, I mean compressible material, we can see that alpha and kappa should be equal. in order to have this expression equal to 0. It means that if we have the same degree of anisotropy in stress and material, we have minimum energy also. We tested this on a patch and a big template, both. And we saw that for a patch, when we change the anisotropy direction and stress anisotropy, we have a clear minimum on the 0 value for this direction. And also, when we change the material anisotropy and the stress anisotropy, we have a clear line, which is a perfect line if material is perfectly compressible. It's very interesting that the central zone and the peripheral zone and the stem part of the plant and also the boundary between two organs, according to the stress and material anisotropy that is observed qualitatively, they lie very close to this line. And this is very, to us, was very interesting. And if you show these two, assume that you have a certain value for stress anisotropy, there is a unique minimum for material anisotropy and anisotropy direction that defines the situation of the material, that minimizes the energy. And here we started from an isotropic material, which is pressurized. Pardon?
Question.
Yes?
I had a question. What happens if you don't first minimize theta and then do the second minimum? You don't know that you have a global minimum, do you? In your two-variable system.
If we don't,
I mean, basically, you fixed a section in your two-dimension and minimize on that section.
Okay, this is the result of both of them. I mean, you can see that the stress anisotropy, the material anisotropy and the anisotropy direction. Here, both of them are shown. So you can see that this is a global minimum.
So you don't fix either of them and minimize. You minimize directly with respect to both.
Okay, I should think about your question a bit, but there is one motivation for fixing first the anisotropy direction, and that is the dynamic for the macrotubules is very rapid. So that is we are maybe hours in plants. But for adapting the degree of anisotropy takes a longer time. So those are not from the same time scale, at least. This is the minimum answer that I can give. So starting from isotropic template and pressurizing it and looking at the energy distribution or energy density on the template, and suddenly introduce the material with some kind of random anisotropy, direction and degree. I mean, making the cells randomly anisotropic, and also these directions are also random. The same pressure, the same overall elasticity, I mean the stiffness of the material, and then turning on the stress feedback on the alignment, and then adapting the degree of anisotropy, we can look at the overall energy on the template. Okay, measuring the energy when we follow this horizontal scenario. And this is the interesting result. For the isotropic template, we have some kind of equilibrium energy. If we introduce some kind of random direction and degree of anisotropy into the system, suddenly energy jumps up. If you turn on the direction feedback, energy drops below the isotropic. And if we introduce the adaptive anisotropy to the system, it drops further down. And so the minimum overall energy on this template is when we have the feedback both on the direction and the degree of anisotropy. Okay, this is a movie out of this, and you can see that the energy, how the energy changes. Here is shown the material anisotropy is shown, and the final step is when the material adapts itself to the stress anisotropy.
So how do you change? Are you changing the mesh? How do you change from one to the other? Computationally, how are you doing this?
We have I take the derivative of the general term of energy that I've shown respect to strain, and I have an explicit expression for the stresses that include all the parameters that I can play with them. But I achieve always the equilibrium before taking one time step to be sure that I am in the mechanical equilibrium. How much time do I have?
Not much.
Nothing? Not much? Or negative or positive? Negative. Okay, I try to wrap it up. Probably I cannot cover everything. Okay, this is not describing the probably underlying mechanism, but it is certainly interesting from an evolutionary point of view. And then we introduced a growth model to the system. At least I can cover this. This is the old Lookhart growth model from the 60s that relates the growth rate with the stress. I mean, wherever we have more stress, we have more growth. But what's the result of this model? We don't like such a thing, probably. Because everything will be inflated, and if we have some kind of irregularity in the shape or anisotropy in the curvature, it will be cancelled out as the stresses are different in flats or when we have different degrees of intensity on the curvature. So the result of such a model is not very interesting for plants. And this is a stress-based growth model. So we asked, what if we have an anisotropic material and we employ a strain signal for the growth? And this is the strain rate on the same setup as we have seen before, when we have played with the different values of the stress anisotropy and material anisotropy. And again, we have observed that the growth rates that are suggested by such a model are very similar to what it has been observed in the plants. And the highest growth rate is on the stem, actually here, when plants want to grow. And the lowest is in the central zone and specifically in the boundary, that we have almost no growth. So I skip these details. These are just some details about how we have implemented this model, but I can show this. This is the result of cell division and growth. I have said nothing about cell division because we don't have enough time for that, but starting from this kind of template, if I apply a stress-based growth or growth on an isotropic template, I mean, I can have this template as isotropic or use that mentioned model of adaptive anisotropy. And the result of a stress-based growth will be some kind of this, as we expect, just inflation and producing a kind of sphere, growing a sphere, inflated cup. But if we mix this strain-based growth with adaptive anisotropy, we can achieve anisotropic growth out of isotropic pressure. I have applied a normal turbo pressure, which is isotropic, but I have achieved anisotropic growth out of that. And the rest are some kind of investigation of mechanical signals on three-dimensional structures that are we have made these templates full with I mean having all the cell walls field inside of the templates, because all of the other mentioned, previously mentioned simulations, inside them is hollow. I mean, there's nothing inside, because we have assumed that the epidermis is the most important. But we wanted to include the internal layers, and this is an experiment of hypocotyl, the stem of an early stage of the plant, and you can see that when they push against the hypocotyl, microtubules switch from almost circumferential direction to the longitudinal direction, and we can capture this in our simulations. We have pushed here, and you can see that the stress direction switches from one direction to the other, and also bending experiment that you can see, you can follow the stresses. And I can skip the rest of the things that achieving the cell polarity out of a stress direction. And finally, I should say that, yeah, you can go through the conclusions yourself. I don't want to keep you hungry, probably. And at the end, the acknowledgements. This is my supervisor and postdoc in Sainsbury lab, Pavel Korpunski and Andrew Larsson, that are working together with me in Lund, not exactly on the same project, but we collaborate a lot. And people from Meirovic's lab in Caltech we have been collaborating with different experiments. Thank you.
So, in your current models, you assume that everything is indeed happening in the L1 layer, and also in the 3D models, you also only model the L1 layer.
In the 3D model,
right? Isn't it? Because that's one layer of cells, and I wonder what is the evidence that everything takes place in this layer.
You're asking about 3D simulations? No. Well, we call these kind of templates that are 3-dimensional, but just one layer. We call it 2.5D simulation.
No, so my question, I was just curious, what is the evidence that the deeper layers, the deeper tissue layers, don't matter for dome expansion?
Actually, there is not many quantitative experiments done because measurements, including the you shouldn't kill the plant if you want to do some measurement, and the dead tissue with live tissue behave completely differently. So it's not really the only measurements that I have seen are mainly about the growth or the deformations, and also recently on the L1 layer only by AFM they can do some measurements on the elasticity. But there is not, but there is one evidence that is very clear, and which is the epidermis is 10 times thicker, almost 10 times thicker than the rest of the cell walls. And also, this is the boundary between the huge turgor pressure inside the plant and the outside. So it is the most resisting part of the plant against the turgor pressure. So it makes sense to focus on that when we talk about mechanics. But we have included the internal layers. For example, this template, it has 3 cell layers. So you can go deep inside, and we can include more and more cell layers, but it will be computationally expensive.
You showed initially at the very beginning that you were comparing shape elements versus triangular elements. Have you done any error analysis, finite element error analysis on these?
No, we have we have been focusing on we have compared some kind of numerically the results in plain tests show a very, very good agreement. I don't have any number in my mind, but they have been really satisfactory. But and you can
Typically, you're committing errors which maybe you don't with the shape elements, I would assume, and you don't quantify how much you lose when you use triangle element. But I don't know.
It's our plan to include to add some kind of bending energy to our triangular springs or the plate elements. We are not claiming that it's perfect. It's obviously it has this weak point of lack of bending energy, but we think by comparing the results, we have been happy of having at least capturing the qualitative behavior of the. But no, we haven't been really investigating that really rigorously.
Any more questions? Thank you, and all the other speakers.
Thank you.